Hi, everyone. Welcome back. We'll continue with webinar 2 now that will be taught, or you'll hear from both Michael and me throughout this webinar series. So this webinar will cover what we need to think about when preparing a combined EEG-fMRI analysis. Specifically, this webinar will be focusing on what we need to consider during the pre-processing. So I am going to begin with what factors we need to keep in mind when performing EEG-fMRI analysis and provide an overview of the different methods for integrating EEG-fMRI data. Then I will introduce the experimental paradigm that we have used to collect a demo data set, which will be used throughout the next 2 webinars for practical demonstrations. The remainder of this webinar will then focus on pre-processing pipelines for both EEG, which will mainly be covered by Michael, and then the fMRI pre-processing, which I will cover. So let's begin with what we should keep in mind when considering a combined EEG-fMRI analysis. You have already seen this slide earlier, but recall that the nature of the EEG and the bold signal is quite different. The bold signal indirectly measures an increase in neural activity through an increase in metabolic demands. And we should keep in mind the neurovascular coupling and subsequent slow hemodynamic response. On the other hand, EEG measures the electrical potential differences as voltages on the scalp that are generated by synchronized neural activity from underlying sources. And due to volume conductance, we measure mixes of sources at the sensor level. Therefore, for both signals, we should keep in mind that other factors influence variation within the signal, and that we also should consider the lag in time between the neural response observed in the EEG data and the ones observed in the bold data. We should also consider that simultaneously acquired data has different dimensions. The complementary characteristic of EEG and fMRI also means that both data sets have different spatial resolution and different temporal resolution. In the data analysis, we will have to restructure the data sets to reduce the dimensions in either the spatial or the time axes of one or even both data modalities. And this is something that we already want to keep in mind for the pre-processing. Now, this paper here shows a nice overview of different approaches for integrating EEG and fMRI data. The image is quite busy, so I'm going to try and break down each section individually. Typically, we begin by doing the pre-processing, and we do this in the 2 modalities separately. And we will go into more detail on each one of these steps later in this webinar. Then after pre-processing, we need to consider how to obtain features we are interested in. We should have already planned this during the study design phase. However, here I just want to point out some examples of features or signals that we can extract from the EEG and the fMRI. And then we can use this in further analysis. This figure also shows how we can integrate data. Particularly, it shows the asymmetrical integration approaches. In webinar 3, we will cover 2 examples of the asymmetrical integration. One example will include taking an EEG feature of interest and using this to make some kind of prediction on the fMRI time series, whereas the second example that we will show will involve using spatial information from the fMRI data and using that to spatially constrain source reconstruction in the EEG data. It's also possible to extract either an EEG feature and fMRI feature separately and then just make a direct comparison. However, we won't focus on that today as it's not really considered an integration method. Then there are also symmetrical integration approaches. These can be divided into model-based and data-based integration methods. In a model-based integration, we have some kind of neuronal model or model of complex neurovascular coupling, and we use that to predict activation within the combined data sets. This does require knowledge of how we expect the two signals to behave within our model. Whereas in a data-driven approach, we do not need to model the complex coupling between the EEG and the fMRI signal. Rather, we see what elements of the signals covariate or couple through information obtained from the data analysis. There has been an increasing interest in recent years in the symmetrical analyses, particularly with an increased interest in using machine learning methods. However, these methods tend to be more complex, so we will not focus on them in today's webinar series. They are worth mentioning, though, for the complete overview. Which type of integration analysis we plan to use depends a bit on factors such as what kind of a priori knowledge we already have about the signal of interest in both the EEG and fMRI modality, but we also need to consider how much artifact will be present in the data and whether we can successfully extract our feature of interest with the planned analysis method. And here we should consider that with some of the more advanced analysis methods, there are various steps that we are doing. And if we have a large number of artifacts still remaining in our data, this will impact every single analysis that we plan to do further along our pipeline. We also need to consider whether the signal in the two modalities truly reflect the same neural response, such that we can use information from one modality to either inform the other or to make more of a direct comparison between the two modalities. I would like you to keep these points in mind during this session and think about some of the assumptions that we are making about either or both modalities and how this may impact the results of an analysis. Now, I'm going to briefly introduce the experimental paradigm that was used for the data that we will do some demonstrations on. It was based off of a study by Porcaro et al., and they showed that when you present a checkerboard, it evokes both a visual evoked potential in the EEG, which could also be source localized to occipital or visual areas, and we see a bold response, again, in visual areas in the fMRI data. Furthermore, they showed that the amplitude of the visually evoked potential and the bold response scales with contrast of the checkerboard. So here in the solid line, we see a high contrast checkerboard versus the dotted line is indicating a low contrast checkerboard. And we can see that both the amplitude in the bold response as well as a specific component of our visual evoked potential scale with the contrast of the checkerboard. So we created a similar paradigm that involved showing either a high contrast or a low contrast checkerboard, and the checkerboard was presented for 1 second overall with a pattern reversal occurring at 500 milliseconds. We also only showed the checkerboard on one hemifield, or in this case the left hemifield, to kind of concentrate the activity on one hemisphere in the analysis and make a comparison more easy. Then one thing I'd like to point out here is that we randomized the interstimulus interval between 17 and 20 seconds. If you're coming from more of an EEG background, this is quite a long interstimulus interval, but we had implemented this in our paradigm to allow attenuation of the hemodynamic response. There is also a vigilance task in this task that we won't focus on today, but we use that to monitor whether the participants were still responding to our task and hadn't fallen asleep or had their eyes closed, as it is sometimes difficult to know what your patient or participant is doing when they're placed in a scanner. And we also did the onset of the checkerboard trial with from the MRI volume marker, and Tracy had already explained why we do this in her response to one of the questions, but we will also get back to that later on in the pre-processing. Then we will only be looking at data from one participant today, but 5 sessions were acquired for one participant, which roughly resulted in 50 high contrast and 50 low contrast trials overall. Okay, now after we have collected data, we would then begin with the pre-processing of our data. In this webinar, we'll begin by discussing the EEG preprocessing. I will first, or only briefly give an overview today of the scanner-related pre-processing pipeline, since we have a very in-depth webinar already available on our website. But one of the very first things that we need to do when pre-processing EEG data is to remove scanner-related artifacts from our data. And this begins by first inspecting what our data looks like, or our EEG data looks like, within the static field. So there are particular environmental factors, such as the cold heads in the scanner, that create noise or artifacts within our EEG data. And even after correcting for some of the other artifacts from the scanner later on, the best baseline EEG data quality that we can expect is what we get within the static magnetic field. So we want to know what our baseline activity would look like. Then the next artifact that we have to correct for is the gradient artifact. This is such a large artifact in amplitude that if we do not remove this, we won't see anything else in our data, and we won't be able to do any further pre-processing within our EEG data, or any processing in general. Once we remove the gradient artifact, we then need to remove the cardioballistic artifact. This can be done via either using information from the ECG signal to remove the cardioballistic artifact, or we can also use hardware such as the carbon wire loops to regress out the cardioballistic artifact from our EEG data. And then once we've removed these scanner-related artifacts, we would then continue to pre-process our data like we typically would do in a lab setting. Now, I'm going to only briefly highlight what this artifact looks like in the EEG data and what we hope to achieve once we've removed the artifact in this data, just to give you a better idea of what to look for in the EEG data when it has been acquired in the MR environment. So let's begin with the gradient artifact. So the gradient artifact comes from the gradient switching during the fMRI volume acquisition, and it induces a very large induction artifact in our EEG data. We can see the gradient artifact here for one particular channel in black, and the data in red indicates what the EEG data should look like once the gradient artifact has been removed. So this is just a very nice example of how large this artifact actually is. We can remove the gradient artifact via an average artifact subtraction method, and this can be done since the gradient artifact is time-locked to the onset of the fMRI brain volume that are being acquired over time, and it's a very predictable artifact. So it is very easy to create an average artifact template that can then just be subtracted from the data. This does, however, require that we have synchronized data between the EEG recording and the scanner clock, and we also need properly timed fMRI brain volume markers. Now, note that if your experimental paradigm is time-locked to these volume markers and subsequently the gradient artifact, and now you average out that artifact, then you run the risk of also averaging out the experimental effect of interest by just implementing the gradient artifact correction. And this is why we recommend adding a slight jitter between when you show the checkerboard in this example, for example, and have the acquisition of the MRI volume. Now, here's an example of what residual gradient artifact looks like. We can see residual gradient artifact across all the EEG channels, and it only happens if we either have unsynchronized data or our volume markers are not properly timed. But we can either expect to see equidistant spikes across all of the EEG channels, or it's even more easily visualized in the frequency domain, where we see peaks along our frequency spectrum, and these correspond to the slice acquisition frequency and its harmonics. Once we've hopefully successfully removed the gradient artifact, we then have to deal with the cardioballistic artifact. The cardioballistic artifact is an induction artifact due to motion related to the cardiac activity within a magnetic field. And there are two methods for removing the cardioballistic artifact with an analyzer, which I will cover today, but there are also other methods available in the literature. So the first one that we have implemented in Analyzer is we can use signal from the ECG, And particularly what you can see here is that this cardioballistic artifact within the two occipital EEG channels corresponds with the largest distortion or the T wave in the ECG signal. So we can use the features of the ECG signal or the R peak, for example, to make a pulse artifact template that we then average together and subtract from our EEG data. What we should then get as a result is the data here in black. The second option involves using carbon wire loops. This does require having the carbon wire loops that we can see integrated on this cap here to record motion-related signal within the magnetic field, and then we can use a regression approach to remove that artifact from our EEG data. Note that if we have residual cardioballistic artifact, which can often happen with option A, then we expect that to impact further EEG analysis in the frequency range below 15 Hertz. Now, we've already heard this a few times today, but movement within a magnetic field induces an electromotive force in conductive materials such as our EEG cap. And this then gets picked up as voltage changes in our EEG recording. And it's a bit difficult to remove spontaneous head motion since we don't really know when to expect it, but we can use hardware such as carbon wire loops that record a reference signal of just motion within a magnetic field, and this information then can be used to remove it from our EEG data. So in this example here, we have a set of carbon wire loops that have acquired some motion, head motion within the magnetic field, and we can see that this artifact within the carbon wire loops looks very similar to what it does in the EEG channels. And if we now implement a regression approach, then we can remove the motion-related noise from our EEG data. And in red here, we see that the head motion has been, or that artifact has been attenuated out of our EEG data. Once we've removed the scanner-related artifacts, we would continue with the EEG preprocessing, and this will now be covered by Michael.
Thanks a lot, Celia, for starting us off. I'm taking over with the screen share now. 1 second for that. And then we continue. There we go. All right. Okay, so I will now take over for the EEG processing part after everything has happened that Celia has already mentioned now. So this is maybe also a bit more interesting for those of you who are coming from the fMRI side and might not be so familiar with EEG processing. So first of all, we would usually start our EEG processing flow with some data pre-processing, which means basically that we are going to prepare the data by inspecting the data and adjusting the data. By adjusting, I mean things like taking care of the channel setup or maybe of the markers. But the larger portion of this would rather be to clean the data so that it is ready afterwards for the analysis that we want to put on top of this. In our example data set, this will be an ERP analysis with the aim of extracting an EEG measure like ERP amplitude or latency. However, it could of course be something else in case that we are aiming for a different kind of EEG analysis like a time frequency or frequency approach. This would then be done on the single subject level for all of your participants' data sets, and then you would bring the whole thing to the group level. However, there might be some feedback loops in here because if on the group level we are still seeing some data that is coming from artifacts in there, you might have to go back onto the single subject level and maybe revise pre-processing. In the next step, you would aim at generating some results, of course. So extract some measures that you can include in a report that you can compare between groups or conditions. Of course, you might want to create visualizations and extract some data for later statistics. You would then also usually automate the processing pipeline so that not all processing has to be done manually for each participant's data set. Okay, let's focus a little bit more on the pre-processing part in here. This is that was a mistake, sorry about that. So that's the part that we are more interested in this session of our webinar series. So let's talk about pre-processing of EEG data. So what is pre-processing and why do we do it? Because the data needs to be brought to a form that is appropriate for whatever analysis we plan afterwards. And this is because EEG signals are often contaminated with signals of no interest. For once, those might be physiological signals. Probably every one of you who has done some EEG is familiar with artifacts from EOG, so like blinking and horizontal eye movements, but there might be other things like pulse artifacts or maybe muscle artifacts. But there is, of course, also non-physiological artifacts that we should keep in mind. And of course, in our setup that we are discussing here, EEG-fMRI, Celia just mentioned that we have special artifacts here, like the gradient artifact, which we see in the sketch. And in this case, special processing techniques have to be used in the beginning to get rid of these artifacts so that we can then actually start off with our normal EEG processing. So the pre-processing is an intermediate process that helps us separating task-relevant neural signals from random neural activity, and it facilitates our path to our analysis goal. And here I would focus also a little bit today, because it will be more interesting for the fusion analysis, on getting to a reliable ERP component with a good signal-to-noise ratio, and also being able to extract features from a desired domain, in our case, scalp domain, EEG or ERP data from single trials. Here, just another sketch to actually visualize the whole idea behind this. So on the left side, we have an ERP with pre-processing. On the right side, we have the same ERP without pre-processing. So you can see right away this looks slightly different. I just want to highlight a few things to note here. So first of all, the y-axis is very different because this ERP without pre-processing is off-center. It doesn't have a mean of 0. Then also there's some high frequency noise kind of riding on top the ERP wave here. Also the topography looks very different, and also the peak of this ERP. So you see this based on this shaded area here in both sketches. This is displaced, so it's not at the same time. So if we are talking about features of an EEG, of an ERP that we want to extract and make use in a different analysis, you can see how no pre-processing might really affect those features, and you will end up with different values than if you do pre-processing to your data. Okay, now I want to come to the example pre-processing pipeline that we actually have prepared because we have some more things to cover. So we will not do a live processing. However, I will talk you through this prepared processing pipeline and just add some information to each of the steps, and also mention a few things that might be interesting specifically for this EEG-fMRI setup. Just to mention, in this pipeline, as Celia and Tracy mentioned already, the MR artifact related processing has already been done. So the data has been treated with those techniques and exported. So our raw data actually does not contain the artifacts anymore. I have just included this one processing step in the beginning here as a placeholder to remind us that something else had to happen beforehand. And then comes the pre-processing part of our pipeline, which has the main aim of cleaning the EEG signal and prepare our ERP analysis. And then maybe also it can be used to handle some residual MR-related noise. Before stepping into Analyzer and looking at this in more detail, I just want to mention that this pre-processing pipeline as also the pipelines that we see later. They have been specifically designed for this specific data set, for this paradigm, but also for this recording to deliver a good result. So you should please not consider these steps and what we have used as parameters here as a gold standard. This has been curated to fit this paradigm and recording. Okay, let's now step into Analyzer. I'm switching over. And here you see now actually exactly the same steps that were just visible on the slide. So let me just walk you through these. So again, this first one is just a placeholder. If I go in here and I show all of our channels, you can see actually there is still some residual artifact here in the beginning, but then if I go in the data, this is gone because it has been treated. What we see here are all our scalp channels. However, there's also some additional channels like our ECG, and down here we have the signal of the carbon wire loops and also some respiration signal. Then we have otherwise data that looks actually quite nice, I would say, but still we should do some treatment of the data. And I just want to highlight that we have here and there some rather characteristic bumps that look like blinks, and they're also in frontal channels, so this makes quite a lot of sense. I also want to show quickly to you, if we right click here and go to the properties, we can actually see that we have 69 channels in here, and our sampling rate is 500 Hertz. So this is different from the usual 5 kilohertz that you would use for the recording, because we have already downsampled the data after doing the MR-related artifact corrections. Okay. So this is the first step. Let's now look into the second step. This is done by edit channels, which you can find under transformations, like all of those steps coming here. And it's here in the preprocessing group in the front. So edit channels has been used here to remove some channels that we don't need. So if we double click here actually and visualize, you can see that now the respiration, the ECG, and the carbon wire loops, they are gone. And this is because we actually are not interested in our example in these, so we do not want to check the respiration or the ECG signal, and also the carbon wire loops, they actually have done their job already by using them in the carbon wire loop regression, so therefore we can just drop these. If you follow me into this menu, that's the right click menu, where we can check a lot of things in the node. There is this edit parameters reprocess node. I will use this now to reopen the transformation, and I am going to use this a few more times in this session. However, I am not going to reprocess the node. I just want to show you the parameters that we have used in this pipeline. So here you go. In edit channels, you can do a few things. You can change the name of a channel, you can change coordinates, you can change the unit, but you can also simply untick channels and then they will be dropped. And this is what you see here. So we simply mark or unmark a channel and then it will be dropped from the data set. I'm clicking cancel again because I don't want to change this now. Okay, so this is the first step, and then comes the second step with a very similar name, edit markers. You also find that below edit channels here in the menu. So now this is about editing markers, of course. So we should look at the markers. You can see them down here below our data. There are a lot of R128 markers. Those are the markers coming from the scanner. But here and there, you also see those red markers, S3, S4, for instance, here. Those are our stimulation markers. And if you're looking down here, you see actually also a lot of blue bars here in the navigation. So this is because we have so many R128s, which are blue, that we only see those. And the red ones, I hope you can see that from the image, in between there. But what you also see now is where there are no blue markers, those are actually the breaks between our 5 acquisitions in the scanner. So if you now again go to the right click menu and go to markers, you can actually see how many markers we have here. And you see now in the edit markers mode, where I opened this, we have renamed those markers. So before we had those numerical names, and now we have here these proper names like high contrast and high contrast reversal, low contrast reversal. And this is just because it's easier for our following analysis. It's just easier to keep track of what you are using if your marker has a very clear name. And you can also see how many markers we have per category. Okay, so these first two steps I would rather subsume under we are adapting our data set, our layout, and now we're coming to the actual steps of cleaning the data. The first one is filters, something that is very commonly done. And again, I'm just right-clicking here to reopen the menu so that you can simply see what we have used. Basically, the message is now that we have dealt with the MR artifacts, we have EEG data, and this EEG data might maybe be a little bit noisier than in our lab experiment, but still you could use now the same processing steps as for a lab experiment with the same paradigm. So if you're now looking to filter the data, you should maybe use the same filter parameters as those that you would use in the lab for the same experiment, aiming at the same ERP component. So we used here 0.1 to 40 Hertz as a bandpass filter, which is a rather common thing to use for ERPs. Just to highlight a few more options here. So we have the order that you can also add for each of those cutoffs, and this is the roll-off of our filter response here. So if I change this from 8 to 2, you see a much less steep roll-off of the filter response, and we are going to use this steeper one. This will create a nice cut, especially as you can see above frequencies above 50 Hertz. So if we still have maybe some high frequency noise remaining from our scanner artifacts, this should actually also maybe deal with those high frequencies. Another thing I want to highlight here is the store data and cache file. This is an option that we generally recommend to use with the filters module because this helps preventing transient filter phenomena. So you should simply keep this active. Okay, canceling this again. If we're now looking into this node and again visualize all channels, you can actually see this already looks very nice. There is not too much high frequency stuff going on if I'm clicking around in the data here. Still, we have those bumps that we can probably relate to our blinking, but we will treat those later. So the next step in our pipeline here is this new reference node. So the new reference is here. Actually, I didn't mention before, the filtering is here. So the new reference, that's a very common step to re-reference your data. Our data has a certain reference from the beginning, and this was FCZ in this data. So if I actually mark some data here, and do a mapping view, you can actually see that there is a hole here in this electrode layout where FCZ sits, because this is the electrode that we are looking at. So the reference is not included in the layout of the electrodes, and therefore there's just a hole basically. Changing from this online reference might be a good idea, because generally we want to use a reference for our data that is far away from the area of interest. So we're looking at visual potentials here. That means like occipital, parieto-occipital areas are probably of interest. What you also often use is something like a mean mastoid reference that might maybe be a little bit too close to occipital areas. What we decided to use in this data set here is a common average reference. And let me just show you how we implemented this by opening the node again. You can see here that we selected all channels to form the new reference. And this is because only scalp channels are left at this point. We dropped the non-scalp channels in a previous step. We also include the implicit or online reference, or FCZ, into the calculation because it is a scalp channel. So it should form also, or should be a part of this newly formed reference. In the next step, we would then apply this new reference to all other channels that we have. And finally, we could also mathematically regain our FCZ channel through this reprocessing step. Okay, so a common average reference is something that is very commonly used for ERPs, also for visual ERPs, it's fine. It has also another benefit when especially doing also this recording in the scanner. So generally, you should avoid including some bias into your reference, which is why you usually use electrodes on the midline or the mean of the mastoid, so to avoid hemispherical bias. And this can also be avoided with a common average reference. But also, if you're lying in the magnetic field with your EEG cap, the effects on the different electrodes could be differential in different places. So therefore, using the average of all electrodes should also make sure that no bias introduces the new reference via this route. So this is something rather nice to do. Okay. Then in the next step, we are actually doing some proper artifact detection now. So this is raw data inspection, which can be found here. Raw data inspection allows us to operate in different modes. So let me reopen this again to showcase. So the first selection you have to make is which method, manual, semi-automatic, or automatic. We usually recommend using the semi-automatic mode because this gives you the benefit of using objective criteria at first, running them across the data, getting artifacts marked, but then you can still interact with the data by visual inspection and also mark additional areas as bad. And also, if you want to, you can also remove artifacts that have been automatically marked before. We have done this based on all channels, and we use the following criteria here. So we use the gradient. This would help with rather sharp edges like spikes, so very sudden increases of amplitude. Then we have used the max-min criterion. This would help with large amplitudes in a certain time interval. So this would be good if really big deflections happen, like when there is movement, for instance. And then we have used low activity. This would be effectively to prevent flat lines. Well, not prevent them, but mark them. So flat lines that could potentially stem from amplifier saturation clipping. And what we have not used at this point is the amplitude criterion. The reason is that this is dealing with absolute amplitudes. And at this point, then, even though we filtered the data already for slow drifts, there might still be some offsets in the data because it's a rather long recording. So I would not want to use this at this point, but we will come to that later, actually, when we talk about additional artifact rejection on the trial level. Okay, I'm canceling this one here again, and then I'm actually opening the raw data inspection node. So the first thing you see here is this red shaded area. That's the so-called bad interval. So that's something that we marked in the data. You cannot really see them down here in the navigation bar because, again, those scanner markers overshadow everything, but I show you a little trick. You can right-click here and deselect all marker types, and then I select only the bad intervals, and now you see where they are. So you can now jump to them, actually, and review them again, and you see, actually, I hope you see that this is big enough on the screen. So you see there are actually a little bit more bad intervals towards the end. And this is because, here's actually a good example, you see the data got a little bit noisier with the end, which is something not uncommon if you're doing several runs, that it's getting a bit noisier the longer the recording takes. I'm just getting all my markers back, so deselect all and select all. Another thing that I want to mention here is that we have here in between those blocks the breaks, and those I have actually marked manually. So I just draw a bad interval over them in the raw data inspection with the mouse, and they come out as these bad intervals. And the reason is simply if there is a break, our participants tend to move a little bit, stretch a little bit, maybe move their face a little bit. So usually there is more noise in these portions of the data. And while we usually don't analyze these portions of data later, there might be some processing steps affected by this noise. Therefore, it's better to mark them with a bad interval. Because the bad intervals, they remain in the data at this time. You could drop them later when you cut your signal down into trials. However, other steps like our next one to come, the ocular correction ICA, or also the normal ICA that we have in Analyzer, or the ocular correction based on regression, and also average, they will actually ignore that intervals in order to avoid introducing contaminated data into your analysis. Okay, yeah, let's actually come to the last point in this pipeline here, which is the ocular correction ICA. So as the name implies, this node is there because we want to treat ocular artifacts. So just showing again all data, and I'm going a little bit inside the data, and actually you don't see the blinks anymore. So this worked quite well. You see actually a blink marker, but no blink anymore. Let me, in this case, actually open something else, not a dialog, but the operation infos, something that you have an analyzer for each processing step. This provides now an overview of the parameters, but also some other useful information. What we used here with the ocular correction ICA is the Infomax restricted algorithm. So that's very commonly used for ICA-based ocular correction. We also use some electrodes as vertical and horizontal EOG activity channels. This is actually not part of the ICA. So our ocular correction ICA has been designed specifically with some algorithm around the ICA that compares the independent components to your ocular channels and indicates which components might be rather vertical EOG activity and which might be horizontal EOG activity. But yeah, so this happens around the ICA. And if you were to use not our ocular correction ICA, but our standard ICA, this is actually missing in there. So since we have not any EOG channels in our layout, as you can see, which is also not a necessity for ICA-based approaches, we are using FP1 and AF7 and AF8 because those channels simply show nice vertical and horizontal EOG activity. And what I did then was basically compute 63 components. And as you can see, this is a nice quality criterion. It converged, and it did so in 101 steps of a maximum of 512. So that's quite good. And in the end, I removed 2 components. And this is because I only used the ICA here to get rid of EOG-related components. So I did not try to remove anything else. However, potentially you could, because you can also use this ocular correction ICA to remove other components that are related to artifacts if you just happen to find them and the ICA nicely isolates them. So something like persistent muscle tension is usually picked up nicely as an ICA component, but also, and this might be a nice feature if you have EEG-fMRI data, if you have residual pulse artifact in the data, this could also come up nicely as a component because this is usually persistent over time. However, this was not really the case in our data here, also because we used the carbon wire loop regression as a pre-processing method, which is rather good to get rid of the pulse artifacts. So let's actually just look at the results here. So I'm actually going one step back now, and I go somewhere in the data. Here by chance, I found a portion which has quite some blinks. So let me now do something nice in Analyzer. I'm clicking on the node with the ocular correction and drag and drop it inside here. And now I have an overlay. So now let me actually zoom in a little bit. So I just show less channels. And now you can see those artifacts that we had here for the blinking in those FP1 and 2 channels in black. Now in red, that's the ocular correction node. They actually have been reduced by quite a lot to like completely almost. So this is quite nice. We have achieved our goal in that case. So you can see also our participant blink quite a lot here. Don't forget, this is a flickering checkerboard task, so something that might rather lead to a blinking a little bit more than maybe some other visual stimulation. Okay, and yeah, with that, actually, I have already gone through all of the pre-processing pipeline. Let's actually go back now to my slides and just summarize this. So first of all, I want to say this was just very brief now, covering all of the pre-processing. And if you are interested in more details about all of these steps, you should really visit our webinar channel, because we have webinars covering everything I just touched. So specifically, recent webinars dealt with EEG artifacts and handling them, and or pre-processing, so designing your pre-processing pipeline. We also have something about ICA analysis, and also, which is nice because we are looking at ERPs later also, there is a special ERP webinar as well. And if you're new to Analyzer, we of course also have an introduction to the software. So the link to our webinar channel and also to our general support and resources section, where you can find additional materials in text and video, is in your handout. Okay, let me just summarize our pre-processing pipeline, specifically based on this EEG-fMRI setup. So I just want to repeat what Celia basically already mentioned in the beginning. The MR artifact-related data cleaning has to come first, because otherwise you will basically not see any EEG and can't deal with it. So therefore, it's also important that the sampling rate is only reduced after this has been done. And then afterwards, you can just deal with the sampling rate that is fine for your EEG analysis of choice. Also, channels that are no longer necessary because maybe they have fulfilled their purpose, like the carbon wire loops or the ECG channel, they can be discarded. But this is really up to you if you want to keep them or not. Then it's basically about applying the pre-processing pipeline of your choice that fits the paradigm and the analysis goals. So it can be generally the same as an equivalent lab recording with the same paradigm. However, due to this recording setup, the data might just suffer from some more noise, and therefore artifact handling is an important point here. So classical artifact handling steps that we just saw are filters, re-referencing, artifact detection automatically or manually, or ocular correction by means of regression or ICA. There are, of course, more artifact handling approaches. This is just a selection. And here, yeah, you maybe have to tweak them a little bit here and there in order to get a nicer signal maybe for the more noisy data coming out of this setup than in the lab, but generally you can use the same steps. And as I mentioned in the end, ICA, if you're using the ICA transformation or the ocular correction ICA transformation, might also be a helpful tool when it comes to treating residual artifacts from the scanner, like pulse artifacts. And just some general recommendations for the pre-processing pipeline. So we would always recommend When you plan your processing pipeline, look at the literature, look what your peers have done with similar paradigms and what guideline papers say. Also, we strongly recommend using the same pre-processing and artifact handling steps across all your participants. If you vary them across participants, it might be rather hard to argue. However, there are exceptions. So artifact handling could be an exception in some cases where you have just data sets that are much noisier than others. Then it's about thinking, okay, should I discard this data set or can I still deal with it? So maybe you can also come up with some justifications why some data sets have to be handled with maybe a little bit more lenient artifact detection criteria, or why you maybe have to use additional steps like maybe interpolating some channels. We would definitely recommend to check the effect and consequences of each processing step. So don't just accept everything that's done automatically. Check for the quality of your steps applied. Always do a visual inspection of the raw data and also after crucial pre-processing steps, for instance, like after the ocular correction, to really make sure that there is no under and also no overcorrection. And rather important thing, if you compare waveforms, make sure that all of them have the same reference. So the same reference should definitely be applied to all of your data sets that will go into the same study in the end. Okay, so far about EEG pre-processing, and with that I would hand back to Celia.
All right. Thank you, Michael. I hope my screen is going to be shared soon. It's processing. Okay, I'm experiencing some difficulties. Let me try it again. All right. Okay, so now I'm going to continue with the fMRI pre-processing. I'd just like to note at this point that we are an EEG company, so here we will focus more on the theoretical aspect of fMRI pre-processing, and I will show examples within the data, but we won't be doing any kind of practical demonstrations as Michael has just done so. When acquiring fMRI images together with EEG, we typically use echoplanar imaging sequences. And then we get fMRI image data set that is consisting of a 4D data set, which contains 3 spatial axes and 1 temporal axis. The spatial axis corresponds to the pixel values for each voxel, which is the smallest kind of unit that we can acquire within, or spatial unit that we can acquire within fMRI. And our time axes will correspond to the brain volumes that we're acquiring over time for our session. Now, note that each 3D brain volume is acquired in 2D slices. This is typically done in axial slices, and these are acquired across time, which then result in our acquisition time, or TR. And now the fMRI analysis itself will observe the evolution of the bold signal time series within thousands of voxels that have been acquired over time. And more specifically, what we're actually looking at are pixel intensities and their fluctuations in time, as we can see here. Now, within specific brain regions, we would expect that we will see these pixel intensities within given voxels to correspond or fluctuate with our experimental paradigm, such as the presentation of a checkerboard. But as we can see from this data set, there are also other variations within our signal, and some of these variations are due to other properties within the scanner that is not related to our kind of experimental response. And this is what we will handle in the pre-processing. This paper or graphical abstract shows a nice overview of different pre-processing steps. It's taken from the fMRI prep pipeline, and I suggest checking this paper out to get maybe an overview of different toolboxes that can be used for fMRI preprocessing analyses. But what I want to point out here is that not only are we using a set of EPI or gold images in our preprocessing, but we usually also obtain some structural or anatomical image along with potentially also T2 weighted images or field maps. And that there is a variety of preprocessing steps that we have to do both to the anatomical and the functional images prior to doing any kind of fMRI analysis. The two main goals of fMRI processing are to attenuate artifacts. This involves motion correction, slice time acquisition correction, and field distortion correction. And then the second goal is to spatially standardize the fMRI data and improve power of our statistical analyses. And some of the preprocessing steps that we use there are co-registering the functional images to the anatomical image, spatial normalization of our subject or participant data to a standard template, tissue segmentation, and potentially also temporal and spatial smoothing. Now, the order of these preprocessing steps may vary and some steps may be omitted, but this very much depends on the type of analysis that you plan. And today I will be focusing on preprocessing steps that are important for task-based fMRI. Note that some of these are used in resting state as well, but the pipeline for resting state varies slightly. Okay, let's begin with head motion. We already saw today that this causes large artifacts in our EEG data, and it also will cause large artifacts in our fMRI data. So we need to consider that when we're acquiring fMRI images, we acquire the brain within a specific field of view. And we could now assume that this black box around the brain is our field of view. And as we are acquiring brain volumes over time, the box or the brain within that box will shift due to either respiration or as the participant relaxes their head and their head starts rotating back, for example. And that means within a given region, as defined by this red square, we can expect the pixel intensities to vary due to movement of the brain within our field of view that is not related to our experimental condition. Since we assume that the brain remains the same shape as it moves, we can calculate a set of realignment parameters by aligning the brain volumes acquired across time to a reference volume. This is usually the first volume within an imaging sequence, and we apply a combination of movement transformations or rigid body transformations these are known as either translation and rotation, and they occur along the three spatial axes. Here we can then see an example of the 6 realignment parameters that were acquired for our particular data set for a particular session. And we can then use these 6 realignment parameters to spatially interpolate the images without motion, for example. One note here is that we always want to check for how much movement there is within our fMRI images because we can really only correct for motion-related artifacts to a certain degree. And if we see that the overall motion in one axis or in several axes, for example, and let's choose translation or movement within one axis, exceeds several millimeters, we then can no longer really correct for that change. And we should consider maybe removing that section of the data in our fMRI data. If we choose to remove it from our fMRI data, then we also need to consider removing that from our EEG data, as we possibly will combine that data at a later time point. One other thing that we need to consider is that the slices in one brain volume are actually acquired at different time points, meaning the slices are temporally misaligned. And this leads to different time courses in the hemodynamic response. So imagine if we have the presentation of a checkerboard within a given brain region, we could expect a hemodynamic response that would look something like this in blue. Now, if we look at the acquisition of slice 1 versus slice 4, we can see that we are sampling different time points between when we acquire slice 1 and 4, and this results in a hemodynamic response that slightly looks different in shape for the two different slices. And this is an issue for statistical analyses because in the statistical analyses, we tend to assume that the brain volume was acquired at one time point. So we can correct for this by temporally interpolating information from nearby time points to estimate the amplitude of the bold response and its shape at the onset of each volume. And this would then result in a interpolated hemodynamic response function, for example, here shown in the red dotted line for slice 4, that then should look more similar to that of slice 1. Note that the order of motion correction and slice timing correction depends a bit on the order of with how the slices were acquired within your MRI scans, so whether they were acquired in an interleaved or sequential fashion. And you may also want to consider here again that slice timing correction only works for a specific range for your TR time. Now, we also need to consider that different tissues have different magnetic susceptibility, leading to variation within the local magnetic field strength. In particular, in EBI functional images, there also tends to be warping or stretching artifacts along a given axis due to geometric distortions. And this will then lead to pixel variations just due to variability within the magnetic field. One solution for this is to correct for this variation by correcting or taking into account the variation within the magnetic field. And the most efficient way to do this is to also acquire B1 or B0 field maps during the actual fMRI or data acquisition phase, and then using those to correct for magnetic field distortions. Alternatively, if we do not have any kind of field maps, we can use biased field estimations to estimate the field inhomogeneities and subsequently also correct for them. However, I would like to note that we need to consider that EEG or adding EEG equipment to the magnetic field will cause stronger field distortions than if you didn't have the EEG equipment in it. And this may not be able, or not most fMRI algorithms may not be able to handle this degree of variation or inhomogeneity. And something else that we should maybe consider is that the setup of our EEG equipment within the magnetic field also has a huge impact on our overall image data quality. So Tracy already earlier mentioned that the number of EEG channels do seem to have an influence on the inhomogeneity of the B1 or B0 field. So the more channels you have, the larger of an influence you have on the magnetic field. But what we can also see in these examples here, we have a B1 field map, and we see an example of no cap to a 64 channel cap. But in this particular example, the cable trees were pressing against the top of the participant's head, which resulted in more B1 field distortion within the top of the head. And this will subsequently then also influence the brain regions that are more central in this area. At the bottom, we see just the image, or this is just an anatomical image, but we see the same effect of having large MR susceptibility artifacts, again, due to the fact that the cable tree, which you can actually see is located here based off of this slight signal loss in the scalp, was then rooted along the top of the participant's head and exited the head coil in maybe not the best or not in the correct manner. And this resulted into large signal loss here, and it'll also subsequently influence the signal to noise ratio within our fMRI analysis, but it will also influence the pre-processing or the ability to kind of correct for this in pre-processing. Once we've removed artifacts by correcting for head motion and timing and field distortions, the next step involves standardizing the individual brain to standard space. We need to consider that in fMRI or in MRI terms, our functional images are still considered to have low resolution, but more importantly, they also have low contrast. So it's difficult to discern different tissue types, and it's also difficult to really see different anatomical regions within these functional images that we have here. On the contrary, anatomical images are much more, have higher resolution, and they're also more, or have well contrast. So we can see different brain regions much easier in these. And one solution for further pre-processing steps is to then also map these functional images onto the high resolution and high contrast anatomical images in native or subject space. And this allows us to then know where we find ourselves within our functional image, but also helps with later pre-processing steps. And this co-registration occurs via affine transformations that then maximize mutual information between the two images. Once we've co-registered our functional images to the anatomical ones, we need to consider that brain size and shape varies across participants. And if we want to do some kind of comparison across our participants, particularly by comparing spatial regions, we need to bring each native brain into some kind of standard space. And this is essentially what spatial normalization is. So we normalize the native brain by using a set of linear and nonlinear transformations to morph that brain into some standard template space. And the template brain or standard brain also has the benefit that we have a precise mapping of specific brain regions that is done by using 3D coordinates, so it's also much easier later than to do a comparison either across different studies or across different participants. And we tend to normalize both the functional and the anatomical images. Then either before a normalization or along with normalization, we also tend to perform tissue segmentation. So fMRI bold analyses are performed within gray matter. And so we need to distinguish the brain between the different tissue types, which is gray matter, white matter, and CSF. And we can see the gray matter, the white matter, and the CSF tissue maps here. What we do want to consider, though, is that some pre-processing algorithms will perform skull stripping and they remove non-brain tissue and then perform the segmentation step. For combined EEG analyses, particularly for ones that are doing EEG source reconstruction, we may want to consider also segmenting skull and scalp tissue maps for then later on estimating our EEG head models. So the scalp and the skull tissue maps have different volume conductance properties, and it can be very beneficial to use the actual tissue maps to better estimate our head models. But we do then also need to consider that if we do have very large susceptibility artifacts in our data, such as this one, it will be harder to segment the different tissue types, and it will then also be harder to estimate head models, for example. Once we have brought our subject's data into standard space, there are a few other things we can do, such as applying a spatial or temporal smoothing to increase the signal-to-noise ratio across our subject comparisons. So smoothing can improve the SNR by further averaging out random fluctuations and noise in the data, and it also can reduce anatomical variations across subjects that could not be accounted for through the normalization step. Typically, when applying spatial smoothing, we apply a Gaussian filter, and this spreads the intensity of each voxel and blurs it across neighboring voxels. The spatial filter width, or the kind of spatial size that we determine to blur our voxels, is usually expressed in millimeters at the half of the maximum value. And here in this example, we can see what the data would look like with no smoothing as compared to applying an 8mm Gaussian kernel. And we can already see that this image looks very blurry here in the functional data, but its impact is better seen when we apply some kind of statistical analysis later and look at brain activation, where if we don't smooth the data in individual subject space, we can see very kind of distributed or isolated like voxels. And then once we've applied the smoothing, the size or regions become larger and we have more larger blobs that now if we want to do a spatial comparison across different subjects, we can hope that these regions will at least overlap to a certain degree. We can then also apply a temporal filter or perform temporal smoothing. This is done because we need to consider that our fMRI time series does not only contain a mix of low frequency fluctuations such as the fold response to our experimental paradigm, but there are also other factors such as scanner drift or even respiration that can influence the kind of overall fluctuations within our signal. And we can deal with factors, or at least typically in task-based studies, we want to deal with scanner drift, and this can either be done by applying a high-pass filter to remove this very slow fluctuating scanner drift from our data. Here we just need to make sure that the filter that we are applying doesn't also happen to match with the onsets of our experimental paradigm. Otherwise, we may also run the risk of smoothing out our effect of interest. But we can also choose to model some of these low frequency fluctuations as confounding effects in the GLM, and that is the statistical analysis that we will cover in more detail in webinar 3. So I will come back to this in webinar 3. Note here that resting state temporal filters do differ somewhat from the ones that we would apply in a task-based design. Now, once we have performed, that kind of summarizes the preprocessing pipeline also for the fMRI data. We now have performed preprocessing both in the EEG and the fMRI data. We have reduced the artifacts within the EEG data, and the EEG data, however, is still mainly processed in what I would call native space. Whereas in the fMRI data, we did also reduce variability and artifacts due to non-neurophysiological signal. And on top of that, we did already standardize our subject space to a standard template. And now we may want to consider how we will combine the subject versus the standardized data for, at least on the spatial domain, when combining our EEG and fMRI data. We also need to consider that we have fewer EEG channels than fMRI voxels, and we still have fewer time points in our fMRI data. In this particular data set, the TR corresponded to 1 second, whereas I think Michael has downsampled the data in the EEG to 500 Hertz, but this still results in a lot more time points within our EEG data than in our MRI data. So one of the next things we need to consider now is how we would restructure the data to then be able to actually perform some kind of integrated data analysis method. And also, as we're restructuring the data, maybe begin to think what kind of features we would extract, and whether we will extract these features just within a specific region, for example, in the fMRI, or just from a specific channel or a specific time point within the EEG data. To maybe give an example of what I mean with how different or how we can possibly restructure the data, I would like to use this example from the paper by Versich et al. And it showcases how EEG and fMRI data were restructured and then subsequently analyzed in a symmetrical analysis. What this paper did, in short, is assess the mutual information between fMRI functional connectivity and EEG connectivity across different EEG frequency bands. And while there's a lot going on in this figure, I just want to highlight the data restructuring that was done to be able to perform and combined symmetrical analysis. So spatially, the fMRI data was parcellated by taking the voxel average for a given region of interest that was defined by this atlas here. And then in the EEG data, EEG sensor data was source estimated to the same atlas. This now resulted in our fMRI and our EEG data having the same kind of spatial axes or spatial resolution. Then on the temporal scale, the EEG data was downsampled to match the time series or the TR of the fMRI data. So the EEG data was averaged across 2-second blocks or intervals that matched the onset of our TR. And this then also brought both the EEG and the fMRI data into the kind of same temporal resolution, which then allows an easier comparison in the later analysis steps. And we will give more examples of how to restructure and then how to further analyze data in an asymmetrical integration methods. Particularly, we will give an example of an EEG-informed fMRI analysis as well as an fMRI-informed EEG analysis. However, we'll go into much more detail in the next webinar series. I know we are a bit early on time, but I would then like to open up for any questions on this webinar.
Hi, Celia. Hi, Michael. I guess you will be joining us soon here. So thank you both for some excellent presentations. We have a couple of questions. This one came in during your first part of your talk, Celia. So you talked about handling different types of artifacts, but how would you remove vibration-induced artifacts in the EEG that you might see in the MR environment?
Yeah, that's a great question. So I think removing vibration-related artifacts that are specifically induced by the MRI is quite difficult by using analysis methods because we tend to see that these vibration artifacts are not concentrated in one specific frequency domain. So you can't just apply kind of a bandpass filter, for example. Rather, they tend to be spread across different, across kind of the frequency domain. One of the best things to do is to actually try and attenuate or reduce these vibration-related artifacts prior to even collecting data. So this would be more of a data acquisition step, but some other things that can help, particularly when having long EEG cable leads that are running through the scanner, is to consider using tape or some kind of padding to prevent the like vibrational noise along the cables from the EEG themselves. Furthermore, there are many factors such as the vibration from the ventilation system from the scanner that can influence the EEG data, and it may be enough to try and pilot what your EEG data or your scanner like static fields data would look like if you have the vibration on high or as high as it can get, or if you, for example, or the ventilation, vibration, or if you turn the ventilation off and then see whether you're able to remove some of that vibration noise from your EEG data. Yes.
Great, thanks, Celia. That's a really good point. We often focus on what can we do with the data without processing, but there are so many things we can do to make our data better in the first place. So yeah, that's really good advice. This next question came in during your talk, Mikhail, but maybe it's something you could both answer. So I'll start with you, Mikhail. Maybe Celia, you could jump in. But do you have automated or semi-automated pipelines for MR-related artifact removal in Analyzer? And then the second part is, do you have any recordings of doing such analyses? So yeah, Mikhail.
So talking about the artifacts dealing from the scanner, right?
Yeah.
Yeah, I mean, those definitely, like all other pipelines that we have in Analyzer, you can automate them. So you basically do this analysis once manually. And then it's, of course, important that the parameters that you input in this analysis are the same across recordings, which would usually be the case. And then you can, in Analyzer, use the history template in order to automate this across different recordings. So there's definitely some automatic part here. Some things might be rather maybe semi-automatic also, because there are some steps. You can think of maybe if you're doing a cardioballistic correction, there are some parts where it makes sense to really actively interact and look for the right template, for instance. So you might actually have to come in manually for a moment, but generally, yeah, as with a lot of processing, you can automate this and save a lot of time. Yeah.
Yeah, great. Celia, anything to add on automating such pipelines, or whether you prefer not to do that?
Sure. I'm going to kind of approach this question maybe from more of a hardware side. So there are older, well, maybe not older, but more established methods that have been used longer for correcting scanner-related artifacts in the data. For example, I mentioned you can remove the cardioballistic artifact by using information from the ECG signal. This is a very semi-automated process because we have to consider that the ECG signal itself is a kind of physiological signal that fluctuates or varies within our scanning session. So it's hard to kind of automate that with some kind of mathematical transformation. On the other hand, more recently, there have been hardware implemented or developed, such as carbon wire loops, that we can use to remove some of the more variable motion related artifacts, because the carbon wire loops are just acquiring motion related activity, and thus we no longer really need to um, rely on semi-automated pipelines. Rather, we can just regress out our motion related activity, including the cardioballistic artifact from my data, and we don't need to spend time inspecting whether our transformation performed as well as we expected by going through all of the data and adjusting it. On that note, I still always recommend doing a comparison to see how well some artifact was removed from your data before and after performing a specific transformation, but it does kind of automate the pipeline.
Yeah, great. Thank you both. And just to follow up on the second part of that question about any recording. So if you want to know a little bit more about the automation and analyzer that Mikhail mentioned, so with history templates and so on, there are some webinars available. So the introduction to using Analyzer, I will show you that that function. And also what Celia mentioned about the carbon wire loop regression being a little bit easier to automate than something like CB correction, you can find in our handling scanner related artifacts webinar or in our carbon wire loop specific webinar. So there are a few recordings out there that you can take a look at where we actually go through the processing of the data. Okay, let's take a look. I think maybe we take one more question each before we go to the break. So, Mikhail, should the pre-processing steps always come in the same order that you showed? Is that a kind of prescription for pre-processing for the EEG?
No, I wouldn't say so. So as I mentioned, the pre-processing pipeline in this case has been really designed in a way that it's good for this data set and this type of paradigm. So this does not only refer to the steps itself and also to the parameters, but also to the sequence. So this was just what made sense here, but sometimes it just makes sense to do it differently. Sometimes it's also honestly reversible. So some steps are interchangeable in position and you will still come up with the same result. However, yeah, I think it's always important to get a feeling for the data, maybe to do some piloting for the pre-processing analysis with the first few pilot recordings, and also maybe check how others applied it. So often, I think nowadays, more often, the exact pre-processing pipeline and steps are very well described in papers. So it's rather nice to follow this. Yeah, but yeah, I would specifically say in this pipeline that we showed, stuff like changing the channel layout or the markers, there are things you usually do in the beginning, but we could have done them later. That would be fine. While, for instance, the ocular correction ICA, it is really profiting from having nicely filtered data, from having big artifacts marked as bad intervals. So it should not come at an earlier point, actually. If we now take the filter and the new referencing and swap them around, that, for instance, might not make such a big difference. So it really depends is the answer, but I would generally not recommend to just from this pipeline, as also from the other pipelines that we presented in other webinars, don't just take them as a gold standard and use them always, but really make up your mind following like developing the right strategy for the data you have at hand. And I would really recommend in that respect to watch our webinar recording for pre-processing, because there's actually a part in there that deals especially with this question. So you will find that link in your handout.
Great, thank you. And yeah, just a reminder to everybody, you can just keep typing your questions in the questions pane about any of the topics that we've covered today. So just keep the questions coming and we will try and get to as many of them as we can. I would just take one more question, and this time for Celia, but it's also still on pre-processing. So there were quite a lot of pre-processing sets for the EEG and for the MRI data. How would one assess whether the pre-processing worked? So how do you know you've done the right thing?
Sure. So maybe to begin with the EEG data, Michael already actually showed some great ways how you can do it, particularly also in Analyzer, that you can just do an overlay of the before and after and just look for the particular artifacts within the time series. But one nice thing also for many of the more scanner-related artifacts that we have is to maybe perform an FFT on sections of the data, for example, and look at the frequency spectrum. So we can also often, at least for the gradient artifact and also the cardiolistic artifact, see whether we were able to remove that artifact well by looking at the frequency data, and that just tends to be a bit more sensitive than looking at the kind of time series data. And on the fMRI side, I think one of the most important things is to also, as Tracy mentioned in her talk prior to doing any pre-processing, is to maybe just scroll through your data and see if there's any kind of visual artifacts that you already see in the data. This could be some kind of streaking or weird ghosting artifacts. And then maybe ask yourself what part of the system could have created that. Is it the introduction of the EEG equipment? Is it due to some other factor or some other equipment in the scanner? Do you see it across all your subjects or just one? And then within the pre-processing itself, especially when we're doing certain steps such as the normalization steps, we can always look at the spatial overlay to see how well we were actually able to, let's say, extract the brain region or able to overlay our subject data to a template and really just look at kind of a visual comparison in space, for example.
Okay, great. Thank you both. And again, we are perfectly on time, so I would leave it there with the questions.